analysis. Before I have Carmen go on, I just want to cover a few things about today's webinar. It will be recorded, and everybody is in listen-only mode. So the way you'll ask a question is to go to the top of your live meeting frame, and on the Q&A tab, you can enter a question there, and we'll answer those at the end of the webinar. You can only ask one question at a time, and then after your question's been answered, we'll close that question out and you can ask another one. But the limitation of the system, you can only ask one question at a time. The webinar is being recorded and it will be available online on our archives that you can listen to, and that link is included with the webinar announcement. And with that, I'm going to turn it over to National Leader for Soil Quality and Ecology, Susan Andrews, to introduce the topic and our speaker.
Hi, I'm Susan Andrews, and I'm very happy today to introduce Dr. Carmen Ugarte, and she is from the University of Illinois. She got her master's degree in soil biology from the University of Illinois Natural History Museum, working on soil, the ecology of mites as potential indicators for soil quality. And then in 2009, she received her PhD, also from Illinois, and looking at the ecology and biology of soil under organic transition. We were fortunate enough to scoop her up in 2010 to work on this current project that just finished up that she will be talking about today. And the larger project was looking at validating the Conservation Measurement Tool, which is the eligibility tool for the Conservation Stewardship Program. And, well, I think I'll leave it at that, but she came up, we'll just say that she came up with some very unique and creative ways to take data that's already published and compare it with scoring of indicators that are used in the tool. And we are looking at this process that she used for populating the dynamic soil properties database, and because of her experience, we've asked her to join the advisory board for the dynamic soil properties. Carmen, take it away. Thank you.
Thank you, Susan. Well, good afternoon, everyone, and thank you for this opportunity to present to you some of the results that we got in this project. Before I move on into the rest of the talk, I just wanted to acknowledge that, like Susan said, we are not working alone, so we are a team of 4 researchers, and we have Hoyan Kwan and Michelle Wander here at the University of Illinois, and Susan, who has been very helpful through the process, guiding us during all the steps of this project. So you see that here in this next slide, I have a brief outline of the topics, the main topics that I'll be covering in this afternoon. I have an understanding that most of you who are joining the webinar today work for NRCS or are related with the conservation programs that NRCS runs, but just in case, I'll provide a brief introduction into the Conservation Stewardship Program. I will also talk about the tools that we use in this project, tools for data mining and meta-analysis. And I will show the example, one example working with meta-analysis in evaluating soil organic carbon in cropping systems. And at the very end, I will provide some summary statements and some recommendations for NRCS, and also some points that could help us to move this work forward. And hopefully, I plan to use about 30-35 minutes of the time that allows, and then hopefully we'll have enough time and we'll have enough time for a Q&A session. So, so the Conservation Stewardship Program, some of you who, if you work for a long time in NRCS, you are aware that the Conservation Stewardship Program replaced the old Conservation Security Program, and they actually stand for CSP at the same time. So at times this short becomes confusing, but it's one of the federal programs that encourages for the adoption of conservation practices or the maintenance of existing ones. It is a voluntary program, so farmers could enroll in 5-year contracts, and they are rewarded, monetarily rewarded for adopting conservation practices. This current CSP came to be after the passing of the 2008 Farm Bill. Like I said, that came to replace the old CSP. So something that's very important with the 2008 Farm Bill, it was not only that it was mandated this new CSP, but it marked also a radical change in the way that conservation programs were being implemented in the U.S. Since then, the programs have been much broader in scope, in the sense that these programs are rewarded for the maintenance, improvement, or the enhancement of a wide variety of other ecosystem services. And they are rewarding, this specific program is rewarding for practices that are implemented across the country. Also, another important point in this Farm Bill is that it calls for the development of tools that evaluate the effectiveness of the program. So with that, specifically looking into the CSP, we have scientists at NRCS and have worked hard in developing a tool that could evaluate series of environmental services that were being considered in the Farm Bill. So to determine eligibility for entering into CSP, scientists have developed this Conservation Measurement Tool, or like we call it CMT. So it is an expert-based opinion, and it has been developed in an Excel interface, and that helps. It is friendly. Many of you probably have experience with it, and it's friendly of use. You can, through this program, you generate a series of inventories that evaluate the conservation performance in an operation. So after applying and enrolling in CSP through the use of CMT, so we feel like we see this enrollment as a process in which you have in this graph, for example, this graph illustrates how the programs are working. You have management practices on one end, and you have ecosystem services on the other end. And the practices that are implemented on this end up having effects on all the ecosystem services that we care for. So how can we relate these tools on different ends? It's by looking at specific properties, measurable properties. So we have here soil quality, for example. We have, how do we measure soil quality, though? So we have properties like soil organic matter, water holding capacity, and so on. So I see this as, so in here, in this next slide, I listed some of the management properties that we could come across with in cropping systems. So we would have tillage practices, diversity in allocation, intensity of disturbance, and frequency, also the type of amendments that are used. And on the other end, we have all these list of ecosystem properties that the CMP is considering. And in addition, it also considers the use of energy. So this is a very ambitious tool trying to measure or trying to quantify conservation on all these ecosystem services. In the language of NRCS, they're called macro resource concerns, but for the audience that doesn't work specifically with the tool, we label them as icosystem services. So each one of these ecosystem services or macro concerns are further divided into 26 or 27 micro resource concerns. So in this next slide, I have the resource concerns, and the work that we started with, it was looking at the parts of the tool that evaluate soil quality and soil erosion. So tools that could lead to evaluation of soil quality and soil erosion, we have these 4 micro concerns in here, which are measurable properties in the field. They are more directly related to, in the tool, are more directly related to soil quality. However, we all know as soil scientists the importance that all these micro resource concerns have on soil erosion. So we divided, so we had a task on looking at the effectiveness of these tools. So we broke down to basically in each one of the macro resource concerns. So this is an example, a specific example of the type of questions that the tool asks for. Many of you, I'm sure, have used the tool, and this is how it looks into the Excel interface. So when you start inventorying the location and the resources, in this case we have an application that has 2 types of location, one that's a 4-year location and one that's a 3-year location. And it has that they have a combination of different types of grains that they're producing. Some are low and some are high, producing residue-producing crops. And the tool also asks for questions that are related to, that are yes or no type of answers, and also that it asks how the farmer is monitoring for signs of erosion, and the practices that a farmer is implemented to prevent those control soil erosion. There are also questions that relate to nutrient management and the way that the nutrients are being incorporated into the soil and timing incorporation. So in the background, the tool looks like it looks much more complicated than in this slide. But in here, I took some of the scores for soil erosion, for some of the questions that relate to soil erosion and soil quality. So, for example, we have here how the tool would relate to the number of different types of crops that are being implemented within a location. So you would have a grain that leaves low residue quantities. It's weighted as neutral. Then you have grains in a location that leave high residue, and they are more positively rewarded. So the weights go from -5 to +5. Okay, so we have also several other practices in here that relate to the depth of tillage and the timing of tillage and the residue management, and like I said before, the application of fertilizer. So here we have for fertilized or amended systems, we have a weighing, and then we have an additional weighing for the use of organic amendments. Okay. I'll come back to this table and to these categories later on. These are the categories that we could, or the questions in the tool that we could relate and we could start thinking thinking on looking at this question further in the literature. So again, so the big question it was what is the influence of management practices, and by management practices I mean tillage, nitrogen source, rotation complexity on soil organic carbon accumulation. And here I got it also the conceptual framework for the NRCS programs that will move us from the practices. So we have the tillage practices, the amendments and the rotation, how that affects soil organic carbon, and how that ends up giving us an indication of how we're affecting the ecosystem service or the soil quality macro resource concerns. So how do we approach these big tasks? For one thing, it was we thought, well, we could just go out and start collecting samples in all the places where across country where PMT is being implemented. We could clearly do that, but that would be a task that would take us many, many years and would require a lot of resources. So another idea was to, and our second approach, the approach that we took was to go out in the literature and feel like we are doing an experiment of the literature and start collecting data from all the work that has been done on soil organic matter, find studies that work either on like proxy, soil organic matter, soil organic carbon, column organic carbon, and so on. So we ended up going that way, and the approach also suits with the needs that NRCS has that requires results on a timely manner to implement these practices and to calibrate the tool. So for data mining, I just prepared a couple of slides also just set the ground into how this data mining works. So we started looking at peer review articles, and we looked at databases that mainly the Web of Sciences, and then we started searching for keywords like soil organic matter, soil fertility and cropping systems, and and how this was if it was changing. So we ended up setting a set and developing a set of developing a criteria that would help us do some quality control the same way like when you work in a lab space, when you're doing experiments in the lab, we need to control the data that we are inputting into our database. So the first thing we needed to do was to look at studies that were looking at a similar question, that were addressing a similar question we were asking. So in this case, it was the effect of practices on soil organic carbon. Second question we had, or that we wanted to do, it was avoid replication bias. How do we do that? If we are using data from several groups that are across the country, where they have different methods and different ways of collecting data, that would also help us avoid replication bias. Also, we wanted to look at independence of studies. As you know, there has been work on soil organic matter for many years now, and there are several long-term studies and replicated studies across country that where data has been collected for many years. And the problem with this is that if we're using the same data from the same site, it's like we are pseudo-replicating. So we wanted to avoid that. So in our specific case, we kept the report on the most recent publication or the most recent sample collection date, if we were able to find, to look into dates of sampling collection. We also looked at the replication and the sampling design in each niche experiments. This is very particularly important when you're doing meta-analysis. I mean, like in any type of study, you would want to have a replicated study, but in meta-analysis, it is limiting, and we cannot proceed any further if we don't have publication of replication in a study. We also looked at controls. So articles in the literature would refer to controls that the author or the working group have set them as controls. For example, there would be studies where they were looking at the changes in organic carbon from the pre-settlement time to this day. So they would try to find an undisturbed site that is nearby, and then use that as a control. However, like in our case, we wanted to compare everything to conventional practices. So we had to create a mindset where we are not letting us live by the control that the other authors have created, but we are creating our own control. Okay. We also looked at the different methods and there is different kinds of sampling, and there are different approaches also when they looked at sampling depth. So we ended up with categorizing our data into 4 sampling depths. We looked at 0 to 10, we also looked at 0 to 15, 0 to 20, and 0 to 30 centimeters. And one last thing, but that's also very important, was that we looked at the statistics that they were reporting. Generally, we see means that are being recorded in the literature. We were very ambitious at the beginning, and we were hoping that we were going to find information on some measurement of variability and standard deviation and standard error. Unfortunately, and it was a realization of the quality of the data that we as scientists are publishing. We found articles less than 3 or 5% of our articles had measurements on measures of variability, reports on measures of variability, I should say. So at the beginning, we started contacting the original authors and asking for that data, but the response wasn't that great either. So we resolved to move on into the use of different methods, still doing meta-analysis, but taking a different approach to the one we originally had. So at the end, when you have your database, you end up with 2 columns. Basically, you will have a column for your control, and you will have a column for your alternative practices or your conservation practice. So what you do is, you end up, in this case, we ended up looking at the effect, at the response ratio, where we divided everything, we divided the alternative, the carbon in the alternative practices versus the carbon in the conventional practices. And then we did some further evaluation to look at the statistical significance of this response ratio. So we end up with a mean, and then we wanted to see, because we didn't have a measurement of variability, we had to switch gears and use an unweighted type of meta-analysis. So what do you do in an unweighted type of meta-analysis is that you assume that all the studies would have the same variance. And then you do evaluation on these variants based on resampling techniques, which are basically computing-intensive techniques that evaluate or generate the same response ratio several times, and then generate a distribution of the new response ratios that were generated, and then you compare that distribution to the original one that you had. And also that new distribution helps to create confidence intervals. So we can assess the significance of the test by looking at the confidence intervals. So in this case, we looked at a 95% confidence interval. So going back to my original table, again, these were the type of questions that we thought that we could, the ones on red, are the type of questions that we thought that we could find them in the literature and we linked to them in the tool. So we looked at the characteristics of the location, the presence or the frequency of the use of cover crops in the rotation, also the number of species within the rotation. Also, that's very important, especially for the soil quality macro concern. We also look at the residue management and the tillage practices that were used, and the nitrogen or fertility amendment. So just to make, hoping that this is making things clear how we define our treatment pairs or our treatment comparisons. We started with the broad scale, looking at cropping systems in general. So at the beginning, we searched for cropping systems, and then we came out with at least, our study would be kept if we had at least one of these possible comparisons, looking at conventional versus organic, conventional versus conventional with conservation till, or conventional with conservation no-till practices. And then from there, we decided to break down into the components of what a cropping system would have. So we ended up looking, and these are comparisons that the data allowed us to look into the comparisons of nitrogen fertility sources. So we compared everything to inorganic practices and normal applications for a region. And then we looked at studies that had reduced fertility practices, reduced use of inorganic fertilizer. Some studies had a combination of inorganic fertilizer with cover crops, or inorganic fertilizer with manure and cover crops. And then we also looked at the effect of manure and cover crops. And we had a last category that it was the use of cover crops alone. I'm pretty sure I still have it in there, and in the next slide you'll see it. Yeah. So you have cover crops only. And then our last category was looking at the length of the rotation. So most of our studies had a 2-year rotation, and we compared that to the 1-year rotation. In the 3-year rotation, we ended up categorizing the few studies that were from 3 to 5-year rotation. We didn't have a great amount, like I said, but that's why we default into creating only one category in that part. So just to show you the distribution of the data, so the dots in this map represent where the data came from, and you have the division there of the non-resource region. And we, not surprisingly, actually, we found that most of our studies came from the north central and the east farming areas of the country. And most of these operations or long-term studies were looking at grain production. In this part, so like you see the number of dots, like I said at the beginning, we were very optimistic and very enthusiastic, thinking that we would be able to find several studies in the hundreds, basically. When we started our search, we started with over 2,000 peer review articles that we started mining from. And at the very end of this study, we ended up with 55 studies that qualified our criteria. So that could tell you or give you a very clear idea of the type of data that we're reporting. I'm not saying that the quality is bad, but we are just not reporting enough data in our published articles. And so out of those 55 studies, we ended up with about like 260 possible pair comparisons. Okay, but they were broken down by depth, and some depth, I think the 0 to 15 depth was the depth that had the greatest number of 30 persons. So then it was, well, like almost like repeating the section where I detailed the criteria for data mining. It wasn't so rigorous, but we found that that's a very clear indication of the type of data that we're publishing. Also, some of those studies were not in the U.S., and that was also a very big limiting factor because there's a lot of research in Europe and in Australia that we, and also in the plains in Canada that we didn't use for this study. So here in this next slide, I have a very typical plot that comes out of meta-analysis. So in here we have the 4 depth categories that we created, 0 to 10, 0 to 15, 0 to 20, and 0 to 30. And here we have the response ratio. So everything in meta-analysis we're going to compare. In this meta-analysis, we are comparing to a baseline or what we call the control. So everything above 1 would mean that the practice or the system was able to, or was improved the concentrations of carbon in that particular depth. So you could see that looking at these 3 depths to begin with, we have organic, conservation, till, and non-till systems, and we have them overlapping 95% confidence intervals, meaning that they are similarly capable of enhancing soil organic carbon compared to a conventional system. When we look at the 0 to 30 centimeters depth, we see that the treatments or the categories start separating apart. We have the organic system with a mean of about 28% increase versus a non-till system that doesn't show any positive response compared to a conventional system. And the confidence intervals for these actually do not overlap, so that tells us that they are very significantly different. The non-till system, the upper confidence interval goes here, where the conservation till is starting, and the lower confidence interval for the organic system goes here. So they clearly do not overlap. These results are somewhat in agreement. They are in agreement with what other reports in the literature have shown. We have seen the meta-analysis in vegetable systems that were being performed on a global basis, and they have seen around like 7% increase in soil organic carbon with respect to conventional systems. Some others have seen around 18% increase, and that depends on the sampling depth. They were looking at shallow sampling depth. Also, we have seen reports in the literature where the effect of non-till has been evaluated, and we see that there is a good, there's a redistribution of carbon through the profile, and where most of the accumulation happens in surface or in the shallow depth. So in here, and to relate these categories to the CMT, what we did is we created scenarios, or we looked at the possible practices that could be rewarded in each one of the cropping systems. So here we have the questions from the big table that I had at the beginning. It's a summary, and then we have the 3 types of cropping systems. And you see that the complexity changes with each system. You have, in this case, and this would be a scenario for typical systems in land resource regions M and N. So, like for example, in an organic system, you would have a combination of high and low risk crops, and you would have implementation of cover crops within the rotation. Despite the use of spring tillage, they also use other practices that at the end, if you add up all these scores, the organic system has a higher weighting that comes out from CMT. And so like our results are agreeing with what we see on how the CMT is rewarding for the implementation of these practices. When we move forward, when we looked at the effects of nitrogen fertility sources, in this slide I rotated the axis so you would see, so each box represents one sampling depth. And then you have the control or the baseline vertically in here as opposed to horizontal before. And then this is somewhat messy, but these are the categories or all the practices, the fertility practices that the data allowed us to create. Something to note in here is that not all the categories were present at all sampling depths. And the numbers show us the number of studies, the possible comparisons that we found at that specific depth. Okay, coming out first from the cropping system and then breaking down into fertility practices. What we see here to begin with is that we have this very consistent increase in organic carbon by looking at the cover crops, the use of cover crops only. Despite here in this depth, specific depth that the confidence interval is overlapping one, the rest of the systems are higher than one. So that is telling us that cropping systems are increasing carbon based on the depth basis. We also found increases with the use of reduced fertilizer and with the use of manure in combination with cover crops and fertilizer in combination with cover crops. All these confidence intervals are overlapping each other, so it suggests that these practices are enhancing carbon in similar magnitudes. Okay. What it was surprising here, it was to see the effects when we looked at the sources of manure. Unfortunately, our studies didn't have enough studies that would look at composted manure or other sources of compost. But when we looked at the source of, like gluten here at the liquid manure, then despite the use of cover crops, at these two depths we found that there was a decrease of about 20% carbon. These results, especially for the use of manure, have been shown in other long-term studies and in other parts of in other areas of the world. When we looked at in England, or when we looked at the long-term studies in Denmark, we have seen that they relied on a heavy application of manures, and unfortunately our studies, so this led us into some speculation that our studies didn't have enough information on the amount of manure that was applied to the system. So this, we think that this small effect is due to the, could be due to low rate of manure application in the system. Also, it's very interesting when you look at the use of reduced fertilizer. Despite being reduced compared to what the region would be applying, in a way it gives us an indication of over-application of fertilizer because we're still being able to produce biomass and incorporate carbon into the soil. When looking into the similar way as we did before, looking at the practices that the CMT is rewarding. So we see here something to point out. It's like a producer would receive a positive score in here for just using any type of amendments, being inorganic or organic. In addition, it would receive the highest of the scores for using organic fertility practices. We have seen in here these results show that the CMT, that we should review some of the scores in the CMT, and we should emphasize into looking at the sources and the quality of quantity of manure that's being applied. So that calls for probably an additional couple of questions in the CMT, but also like to evaluate this type of to account for these changes. And also, our results agree with the use of cover crops in the tool, the score that is being given for the use of cover crops uses the maximum positive score, and you see here because, like we discussed before, you see the differences, the consistent increases in carbon. So when we look again located in here, the axis back to the original one, or the very first one, and you see the baseline horizontally. And this is the effect of the rotation length. And where you see that the 2-year rotation, based especially in the 0 to 15 and the 0 to 30, it wasn't much better than the 1-year rotation into increasing carbon. And most of these rotations were characterized by a corn soybean rotation or a sorghum soybean rotation. And there has been some indication that the use of these short-term rotations actually do not help in increasing carbon. And then consistently and confidence intervals didn't overlap with one, so we say that the 3-year rotation, which also accounted for the 4 and 5-year rotation, consistently increased carbon to target depth. So for this case, and we found, we looked at the scores that CFT would generate, and here we plotted in this table, we plotted a typical rotation from the more dominant land research regions, and we have a 2-year rotation and a 3-year rotation. A 3-year rotation will have a combination of one low residue and a high residue crop, and it will not receive any, it will probably not receive very high points for the diversity in the rotation. They generally don't have either cover crops or anything like that. So it would add up to, in this section, to a total of 3 possible points. When you look at a 3-year rotation, you have the combination of low and high residue producing crops. But in general, our 3-year rotations had at least 2 crops that were high residue generating crops. And then they have high diversity systems where they would have also cover crops, or they would have some sort of a perennial lay system that is more typical of an organic system. But so that would add up to a total of 11 possible points in this section. So that illustrates that this is in agreement also with the results we found in the meta-analysis. So with that, this is part of, like Susan said at the beginning, this is part of bigger projects. So if you tune in tomorrow, you hear from Hoyang, the other section. But from the meta-analysis, we say that, or we can conclude that the way that the PMT is rewarding for the use of cropping systems is in agreement with the meta-analysis, and we see that all these systems are similarly capable of increasing carbon. We also suggest that when we agree with the report that influences the use of cover crops, however, we think that further breakdown should be done into the use of organic amendments, being very careful to account for the use of to include information on the quality and the quantity of manure application. And that the scoring that's assigned or that's given for the length of the rotation should plateau after 3-year rotation. Also, like we saw that the majority of our studies were concentrated in 2 regions, we think that the inclusion of other equivalent of data from other equivalent biomes in the world could help us to create a bigger data set and could help us to also to make more robust conclusions about this data set. And this is more like for us as scientists that we should try to use consistent terminology. When you mine in the literature, you have no idea the heterogeneity of terms that you find. You think, well, like in NRCS, you have some equivalencies of the depth of tillage and the implements that is being used. However, like for the rest of scientists who publish that data or that type of terminology, it's not encouraged. So NRCS as an institution and us as scientists, I think we should get together and try to publish something that many other people could use later on. So what do we see from here on? I have, well, like we worked in this area first, like looking at the effect of, in the cropland section of the tool, we looked at the effect of soil organic carbon, and this is a summary of the areas that we found data from, and the areas that were more densely populated. And we also think that some of this data could be used for validation. However, we can also use some the models that are available. So Hoyang will talk tomorrow about the modeling part. So we could use like the century model, for example, that could look at the change in soil organic carbon, and that would use information that goes backwards into the historical data in an area of how the land was managed. When we looked at soil erosion, in a similar way to our enthusiasm, the enthusiasm we had for soil organic carbon, we thought that we would have great data set. However, we ended up, because of the heterogeneity on the data, we ended up using the data that was used to work on USDA and Russell that and the data resides at Purdue University. And with that, so we ended up using Russell to the soil erosion evaluation. Some other properties that we could use, we could default to the use of phosphorus to look at the water quality macro concern, or nitrous oxide for using the air quality macro concern. And we have started and we have done some data mining into this. The problem we had with phosphorus is that the method are incredibly variable, and it becomes very difficult to create a control. Many methods look at total phosphorus, other research groups look at inorganic phosphorus, and in very specific areas, and given that the CMP is a nationwide tool, so that makes the task is harder. So I'm not saying that meta-analysis or data mining will not work, but some of the work that NRCS is doing using functions could probably also work for the validation or evaluation of CMP. In terms of nitrous oxide, we have also looked at the literature, and we think that it is a friendly property towards mining. We should also consider, however, other adaptive factors like soil moisture that have a huge effect onto the emissions, into the nitrous oxide emissions, and we don't see that is actually being currently reflected in the tool. Some other things that we can, some other approaches that we could use to validate are, again, models. We can use the daily version of Century, and we can also use OMSDR. And those are ideas of things that we could use. So to continue this work into the validation of the tool. So with that, I think I will just open the board for questions, and I would be happy to hear questions or comments that would help in here. Thank you for the attention.
Well, thank you, Carmen. And again, we'll take questions, and you can do that by entering those in writing at the top of your live meeting frame. There's a Q&A tab. And I'd also take some live questions from the audience here at the National Soil Survey Center with me, if there are any. So, Carmen, our first question looks like there's multiple parts to it. There is much environmental difference east to west in U.S. systems. Why limit research results to the U.S.? How does meta-analysis account for differences in treatment study to study, such as nutrient application rate or source, cash crop used with cover crops, irrigation versus rain-fed systems, etc.?
Okay. So we limited, first of all, we limited the research, the data mining to the U.S. so we could relate with scores that were coming out from the first and the second final CMP sign-up, so CSP enrollment. And then we are very aware of the differences on the climatic region, and that was one of the reasons why you saw in the map that we classed them into the land resource region. And also, that's part of Koyang's work where he incorporated in his models some more region-specific information into the model. So in here, we thought also in doing some regional work, and we would end up limiting our analysis even to a greater extent because we would end up working with the data that is from two main land resource regions. So I hope I'm answering the question.
Okay, our next question concerns the negative scores for fall plowing. Why no negative score for fall plowing?
Why are negative scores for fall plowing?
Why was there no negative score for fall plowing?
I think there is a negative score for fall plowing, and there is also a negative score for spring plowing. However, the one in the pool is much higher. So yeah, fall plowing is just a -5, and spring plow gets a -4.
Okay, I must have looked at the graph wrong. Second question is, soil organic carbon increases as standardized over all soil textures and climate zones and drainage classes. 10% increase is awfully hard to measure in the field.
Can you repeat the question, Sean?
It just says, second question, soil organic carbon increases that are standardized over all soil textures and climate zones and drainage classes. So, interpreting what the question is, should there be differences in soil organic carbon increases between soil textures, climate zones, drainage classes, that kind of thing?
Yeah, we think there should be some differences. And going back to the first question, that would relate to the land resource region. And again, it's some part of the analysis that we could probably go deeper, and we will have to resort into mining for that type of data from other sources and not specifically from the peer review literature. And like we said, like I said before, also like Oyan tomorrow will talk about this specific region-specific data that he could include in the model coming out from Russell 2 and databases, the original databases.
And kind of a programmatic question perhaps, but the next question, if NRCS changes the ranking during sign-up, won't this greatly impact your results?
If the way it's changed, sign up, I'm sure as long as if the rankings are kept, probably the results will not be so much affected. I mean, the primary data that we're looking at in here is data from peer review articles, and we are relating it to recommendations on how the weighting should be changed. So we are hoping that actually our work will help change those scores.
Okay, and then there's an observation here saying we need research on systems in land resource regions A and B. I know you had a table that showed where there was good coverage between the land resource regions, and I think in the central part of the country there was really good coverage.
Yeah, so in A and B, so that would be the north west part of the region. We have some case studies from the region, dependent on area mostly. Some studies that I could recall from there were also hard to evaluate in the sense, or hard to include in the sense that they didn't have information on replication. So, yeah, but I think we had, well, in A we don't have any studies, but in the region B we do have at least 6 or 7 per comparison.
And then, Carmen, our last question is, can we get a copy of the presentation?
Absolutely. I mean, like this would be recorded, so it is recorded, I mean. I don't know if you would want a PDF of the presentation. That would be also doable.
Sure. And yes,
Send it to Susan.
Sure, and if you send me that PDF, I'll certainly include that with the recording of this presentation. And that'll be posted in the next few days. So I thank everybody for your attention and participation. And that will close out our webinar today. Thanks all.
Thank you.